fortunate to have Professor Manoj Verma. He's a colleague of mine and a great scientist, I would say. I have distracted with him for a long time. And okay, so I just talk a little bit about, he completed his BTech in 1999 from IIT Madras, followed by his PhD from Purdue University in 2000. He started in 2005, or he completed in 2005. Then, he was the director of engineering at Prospect at Purdue Research Foundation from 2005 to 2007. Thereafter, he joined the institute in 2010, and he got promoted to associate in 2014, and now he's a full professor. And the work that he does is quite exciting, and that I also talked about in the morning. So it is all related to single molecules and it's quite interesting. And his broad interest is in biosensors, nanobiotechnology, microbiologics. It's a vast field. So I just ask Mr. Manoj to please take over and enlighten us. Thank you.
Thank you. Thank for the opportunity to talk at this forum, and thanks to Prof for organizing this. So the topic that I'm talking about is solid state nanopores for single molecule sensing. So two things I want to do. One is that to talk about the clear overview of this nanopores, exactly the same clear overview of this technology. Now, to show what we are doing in our lab, what we are considering our labs. So that was the two aims for the talk. At any point, if you want to ask questions, free to interrupt me. I do not wait to the. So, of course, those are the departments where we work in. There's the, of course, the IISc, which we are doing, and then there's the department where my lab is located. And so, in this department, there is something called the National Nanofabrication Center, which is South India's probably the largest and perhaps we should say the most comprehensive facility that we have in the country. And it is open to everyone. So open in the sense that you can do your experiments, you can book instruments, reserve instruments, and use these instruments for a fee. Right? So there's a fee structure for usage, and you can choose. And so it contains the instruments that I'm going to talk about, the instruments that we use to make these things. So I work with a fantastic group of students. That's a lab group. And also a set of collaborators who are play a very important role in what I'm doing. So many of them are from SENS. Actually, I is from SENS. Verma is from chemical engineering. She's done in some fellow. Prof is a person in physics who does molecular solutions. Angela Rao was a person who introduced us to this problem. She was a professor at Harvard for a long time, about 30 years or so, and then she moved to this institute that was set up to do more specialized, more focused work on environment. So these are sort of people with whom we have interacted over the last few years. So the work that started in around 2015, 2016. So we've been working in this area for the last 6 to 7 years. So that is the thing. So now coming to what is this nanopore sensor? So if you look at it, if you look at it in the simplest way, describe it as just a molecular scale analyzer, which connects to electrolyte inputs. So now imagine that you have this, so this is one so electrolyte is like something like KCl, NaCl, many of those solids, like salt solution. So you have one reservoir here, another reservoir here, and this thing is completely sealed, absolutely sealed. Right? So the liquid cannot flow between this and this, except that in between these two reservoirs, our nanopore chip sits. And on that chip, you can see there's a hole here. Okay? And this hole is very small. But by very small, we mean that it's of the same dimension of form as single molecules. If you take a single protein molecule or a DNA and so on, these are typically a few nanometers in size. Okay? The diameter of this hole is basically, it could be as small as we can actually make things which are as a true or 3 to 4 nanometers. The dimension of the score is comparable to that of a molecule. So this molecular scale approach. Right? So what happens when you have such a thing? And of course, this membrane that you see here can also be very thin. Although in the picture it shows like a very thick thing, in reality the membrane is also very thin. So what we make today is about 10 nanometers, but we have been working on reducing that to actually again 2 to 3 nanometers. Right? So basically, we have a molecular size approach. Now imagine that you have something on one side, one of these zones, right? So this is called, so normally one of them is called cis and trans. So cis is where you start from and molecules go to the trans side. That's not very important. So we have reservoir 1, reservoir 2. And because these are electrolytes, they have ions in them. Right? For example, if you have potassium chloride, you'll have potassium ions and chloride ions. So if you apply a voltage between the two reservoirs, that's going to make the ions flow. Right? And the ions cannot flow except through this reservoir. Okay. So the only connection between this one and this one is through this very narrow restriction, very small hole. So when the ions start flowing, you will start measuring a current, this ionic current. Now imagine that you have molecules which are in one chamber, and because of the flow of the ions, it basically sets up a flow pattern which is going through the pore. So the molecules will also get transported along with these ions through the pore. And because the size is so small, at any given point of time, only one molecule can pass through it. It's not possible to have 2 or 3 molecules going through simply because the size is so small. So by definition, this structure will act as a single molecule analysis because the only way molecules can go from one end side to the other side is in a single file kind of form. Right? And as the molecules are going through, it will modulate or it will change the flow because the molecule is sitting there, ions cannot block it, right? So because of that, you will actually start seeing dips in your current, and not just like or standard dips, and there may be some shape to the dips also, because the molecule is going to fluctuate. When the molecule fluctuates, the ionic flow will also fluctuate. The idea is that by analyzing this electric current, ionic current signature, can be learned. What can we learn? How much can we learn about the molecule that is possible? And we can use that in various applications like sensing and so on. So that is basically the complete. What we do is to make, we also make these processes and things like that, and then we use optical electronics to do this. Now, when you start looking at nanopores, then you realize that there are 2 kinds of nanopores. One of them is called biological nanopores. The other one is what we work on, is solid. So the biological nanopores consist of single protein molecules, and proteins, as you may be aware, they are these biomolecules which have very characteristic 3D shape. That the proteins will fold into a particular shape. So these are a class of proteins which are called porins. So they actually form a structure. In the case, this molecule is folding in a structure that forms a channel. Right? And molecules can be transported across this channel. And this channel is also of the same dimension of molecules. So this channel is also will be a few nanometers in diameter and a few nanometers. So what people do there is that they take a lipid membrane, and these protein molecules self-assemble on top of the lipid. It's just a chemistry. And so when this happens, so exactly like the way I showed in the previous picture, the lipid membrane can form a seal. Lipid membrane is a hydrophobic thing. So again, it will not allow anything to go through that. So it forms a sealed barrier. But the moment this protein comes and sits on it, the protein has a channel through which things go. So you can have the same way. Nature has already built something like that. So people use this also to make nanopores. And what we have been trying to do is to create completely inorganic versions of this. The reason we want to do that, as I've explained in later slides, is that proteins in general are not very stable. So these, again, as it's the proteins require a particular pH and conditions and all that, so that they can maintain their shape. If this shape gets changed, then your nanopore is no longer effective. It's not wrong, right? So if you make things in silicon and so on, these are very robust materials, and therefore we may be able to make things which are much more resilient to environmental factors compared to what these things are. That's the reason we are working on this thing called solid state nanopores. So today, for example, the biological nanopores is a commercial technology. It's commercially available. There's a company called Oxford Nanopore things where they actually have put single molecules.
So if you take the biological codes, because they are proteins, they fold in whatever manner they fold. I mean, you can't really change it. You can't, for example, make a 100 nanometer protein. Right? Whereas we can fabricate whatever diameter we want. So we can go all the way from, let's say, 3 nanometers to 100 nanometers or 200 nanometers, where we can not only do single molecule, but we can also look at, for example, single virus particles or single bacteria and things like that. So there's a much larger spatial range that we can, therefore we can have integrated sensing thing, maybe going all the way from single cell to single molecule. So such a thing is possible. As I already mentioned, these are likely to be much more stable because we are not using any organic molecules that are susceptible to all kinds of degradation. Finally, there are lots of new 2D materials that are coming. A lot of people are looking and people are discovering new kind of physics which are there in these materials. And right now, the only way people read out these sensors is by using the analytical, which has this problem of not being fast enough. It can only measure things at kilohertz rates and so on. Now, obviously, one would like to do better than that because people are talking about DNA memories and things, storing information in DNA. So when you want to read out that memories, you cannot read out things at kilohertz rate. It's not possible. So if you want to look at alternate readout mechanisms, which are much faster, maybe some of these new physics that people are discovering in these materials can help. And then you can try to integrate that with this one, which are not possible to do with biological code.
Recording in progress.
So that's the reason why we want to do all these things. But of course, to do that, we have to address these challenges. So what are the materials that people typically work with? So as I already mentioned, we want to get things to be very thin. So as a result, that's quite a bit of work that is happening in the Delphi and and so on. But primarily, starting material is generally being silicon nitride. So silicon nitride, the way it works is that we make membranes of silicon nitride. So there's an underlying silicon wafer here, and then you put the silicon nitride, and then we can drill a hole. And I will talk about this in greater detail in the coming slides. So what we do is the following. So we get the silicon wafer. So we buy the silicon wafer. And in the sense building that I showed, as I mentioned, there's a national fabrication facility. So in that fabrication facility, we can do these processes. So the first process to grow low stress silicon nitride film. And so we can grow 200 nanometer in this particular case. So there are various recipes that we have optimized over the years. So this particular recipe, we take about 200 nanometers of silicon nitride and we deposit them. So you deposit it deposits on both sides of the silicon wafer. So second thing what we do is that we do what is called the backside process. So there are two sides of the silicon wafer. It's a kind of arbitrary width which you want, which is backside, which is front side. So there are two kinds of silicon wafers. One is a double-sided, single side and double-side polish. So you have double-side polish. Both sides are identical in terms of roughness and so on. But so one of them is it's the back side, the other one is the front side. So what you do is that you put a photoresist on the back side, and then you have, I guess you are familiar with the concept of photolithography. So what happens is that you put a photoresist and then you illuminate with a UV lamp, and wherever the UV light hits, photoresist chemicals composition structure changes. So it becomes cross-linked or it becomes pure organic, whichever it is. So what happens is that when you then use the development, then that photoresist portion will go away. Right? So you expose this area. And then what we do is that silicon will etch. Silicon can be etched in something called potassium hydroxide. And that etches something that happens in a V-shaped thing. So it's called an isotopic etch. Right? So it starts at one diameter, and then as it progresses, as it goes inside, it keeps getting smaller and smaller. So what we do is that we start that etch process, but before that there's a small thing. I mean, there's also a silicon nitride here, right in the back side. That will not get etched by KOH. So what you do is that we have to first remove that portion using another process called reactive ion etching. So first you use that to remove this silicon nitride, and then use the KOH to remove the entire silicon. So the complete wafer that is there, all of it gets removed. And finally what you have is this silicon nitride film that is suspended. So as I said, we have about 200 nanometers. That thing gets suspended here. And then this is an older recipe. We can use something called electron beam lithography. So again, that works in the way that you basically put a resist material, and then use the electron beam to open a small window. And it works exactly like photolithography. It's just instead of photons, use electrons. And then what you do is that you can use again some RIE or such processes to make a small pore like that. And this will be the final chips will look like this. So all of these processes, they happen in cells. So we have the complete, all the way from the wafer to the chip, everything can be done. Right? And here is a picture of a actual membrane that is suspended. So I mentioned 200 nanometers as a film thickness there, but we can go, obviously we don't want 200 nanometers very thick, right? So we have to thin it down. And alternately we can also deposit very thin films. So we can deposit like 20 to 30 nanometer films also. So what you see here is a very thin, probably around 20 nanometer thin film, and we are seeing from the back side, right? So this side. So a microscope is viewing the chip from the back side. And so why do you see this color? That is because it's something called differential interference contrast, DIC image. So DIC, depending, there is some optical interference that happens. And as you change the contrast, you can see the difference. So we use the DIC to make sure that the membrane is perfect. Because membrane, as I said, is very thin, right? So thin membranes are prone to cracking. And the size of the membrane is also like probably about 30 microns or so. So you have a 30 micron by 30 micron square, which is only 30 nanometers thin. So it's like 1:1000 kind of aspect ratio. So these materials are very fragile. Of course, there are ways to make it very robust. We have done, we are in fact we'll probably going to file a patent soon to make this more robust. But these materials are prone to cracking. So we want to make sure that when you make them, they are not cracked. And the best way to find that is to put this in the microscope with a DIC, because then you will see a very intact area with the same color. Then you know that everything is uniform, it's homogeneous membrane, it is sitting there. So we can make with this process 15 to 30 nanometer nanopores, and we can see that the distribution of nanopores that we make is very tight, right? I mean, like for example, the average here is 28 nanometers plus or minus 2. So we can make them very precisely. Like we can make like 28 nanometers, and if you make another one with 25 nanometers, they are different. Right? So we can very precisely control up to a few nanometers. We can precisely control the diameter, and the diameter is controlled by the dosage of the electron beam and things like that. So that is done. But these are around 30 nanometer thick silicon nitride membrane. But as I said, okay, and so here is a cross section. So here is a cross sectional SEM of this chip. So this is the silicon wafer, and this is the etching. As I said, the etching is anisotropic, so it goes like a V-shaped thing. And the membrane is basically, you can hardly see it because this thickness here is about 300 microns. And this membrane is only about 20 nanometers, right? So you can't even see that in the thing. So it's where it is, right? But as I mentioned, let me see if I have, yeah. So as I mentioned, this is not good enough, right? Because what I talked about in the first slide is that we want to have molecular scale approaches. But we are talking about 30 nanometers, kind of diameter, 15 to 30 nanometers diameter and 30 nanometer thing, right? But that's like, it can fit in a lot of different molecules. So we want to make it even smaller, at least the diameter. So for that, what you do is that we have a transmission electron microscope in sense. So the TEM has very high energy electrons. So in electron, the thing is that the higher the energy is, the smaller the wavelength. Which means that you can focus these electrons to very, very tiny spots. So by focusing these high energy electrons to a very tiny spot, we can even make things which are like, in this case it is 3.8 nanometers. And the smallest we have made is like close to 3.2 or something like that. Right. So using that, we can actually make pores which are as small as 3 nanometers, which is very, very close to what the biologic proteins can do. So at least that part we can do. So now, so that is where we are. So today we have chips which are typically the membrane thickness is from 10 to 20 nanometers is the membrane thickness, and the pore diameter is 3 to 4 nanometers. Right? So that's the kind of pores that we work with. Of course, as I mentioned, we want to go to thinner membranes. That's a different story. But then what do you do after you get the chips? Right? So these are chips which have them. So we put them in a flow cell. So this is like an example of a flow cell. We have made different variants of the flow cells. We also have an optical flow cell where we can, as the nanopores experience happening, we can measure them optically. Right? So in the flow cell is very critical because you need to make sure that there is no leak at all. The current that you are measuring, as I said, is of the order of picoamps to nanoamps, which means that even a very tiny leak of liquid can contribute to some current. Right? So we want to, so basically the leaks are of the order of, the way you quantify the leak is by measuring what the resistance is. And so we are putting a voltage across the two reservoirs and we are measuring the current. So you can come up with a resistance value. So we want resistances that are of the order of gigaohm. Okay. So 100 megaohm to gigaohm. So 0.1 to 1 gigaohm is a kind of thing that we want to make sure that, so that would be a good flow cell, a flow cell that allows you that sort of leak proofing. So that is the thing. And so right now, so again the flow cells we make, only thing that we don't make today is the electronics to do it. So this is something that is a startup from Italy. They have made this very compact device to measure very tiny currents. So these can measure currents down to, as I mentioned, picoamp level. The main thing is that the noise levels here are very, very low. So we want very ultra low noise. So basically it means that 10 picoamp sort of RMS noise. So RMS noise is something that you measure over the entire bandwidth. So the bandwidth of this thing is 100 kilohertz. So across is 100 kilohertz, 10 picoamp is the thing. So if you look at the, another way to quantify noise is the noise power spectral density. Like, so there it will be something like femtoamp sort of, femtoamp per root hertz kind of thing is what you get. So they are very, very low noise systems. So you use something like that. And what you do is that you take AGC electrodes, silver colored electrodes, and you put them into the two reservoirs, and those are connected to this amplifier, and then you can start measuring these things. And so what kind of things do we measure? So we first started with measuring single nanoparticles. Those are the easy things to do. They're the easiest things to do. So we have 5 nanometer diameter gold nanoparticles, and what you do is, so what I showed here is actually the data from that. So here is when there is only buffer, there are no nanoparticles, and now you put the nanoparticles and you start seeing these blockings. Okay. You start seeing this. I mean, they look like neural recordings, right? I mean, if you put this patch clamp and so on on a neuron, you kind of get the spiking train. So it kind of looks like a spiking train, and each of these spikes is basically a single nanoparticle going through. A single 5 nanometer particle going through is what you see here. But how do you know? How do you know it's a single nanoparticle going through? So to confirm that, we do some modeling where we actually simulate the physics of a particle actually going through the pore, and we calculate what is the current that we are supposed to get, and we see a particular shape of this current trace that we are supposed to get. And then you go back to the experiment. So unfortunately this is kind of rotated. So this is the shape, and this is what you see in experiment. So the characteristic thing here is that there is a rise and then there's a dip. And we see the same kind of pattern here. There's a rise and then there's a dip. Right? So these are each single spikes that we have extracted. So then we kind of get the confidence that yes, we are actually measuring single nanoparticles passing through. And then what can you do? I mean, that is fine. Can we do better? So one of the things that we did is to see whether we can measure the charge content of the nanoparticle. So for example, if you have nanoparticles which have different surface charges, can you discriminate them? And this is something important because a lot of times in viruses and bacteria, the charge state of the membrane changes under various conditions. So for example, dead versus live bacteria, and you know, because the proteins around in the membrane changes, surface charge also changes. So we did an experiment where we just switched the charge from positive to negative, and you can see that this is a negative charge and this is the positive thing, and you can see that there's a very big difference in the statistics of events. Right? So what we have plotted here is a dwell time. This is basically the time of the pulse. There's a very big difference in the time of the pulse. Basically, when the surface charge is positive, they take almost 3 orders of magnitude longer they spend in the pore compared to the other case. And the reason is because pore is negatively charged. And now you have this particle which is slightly positively charged, so there is very strong electrostatic interaction which causes them to slow down rapidly. Okay. So having shown, of course we have some model and all that, that's not very important. So having shown that we can do something useful, which is basically measuring the surface charge of particles and so on, we want to go to single molecule level. So, but then the problem of chemical selectivity comes. How do you make them selective? So what we came up, of course, one of the standard ways of what people do in biosensing is that if you take any, let's say silicon wafer or something, there are lots of these surface chemistry protocols where you put this wafer in various solutions, and then these linker molecules will come and sit on the thing, and then you can attach proteins and so on. So people have tried such approaches, but the yield has been very, very poor. People have not really demonstrated the efficiency of those things, and the reason probably, I don't think there is anyone has really looked into why that is so and so on. But one possibility is that because these pores are very, very small, there is a very large barrier for the liquid to go in. There is a very, very strong hydrophobic barriers and things like that exist. As a result, I think that the yield is very poor. I think that's one of the reasons why the yield is very poor. But one of the ways in which we thought could be better than doing that sort of comparison is this approach that we have, which is something like a modular approach. So what we do here is the following. Here is our pore. And what we do is to use something called DNA origami, which belongs to the class of techniques known as DNA nanotechnology. So the DNA origami, the idea is that we can use DNA to make nanostructures of various shapes and sizes and so on. So in this case, we have a rectangular sheet of DNA origami. So the sheet thickness is only about 2 nanometers or so. And in the rectangular origami, there is another pore, there is a smaller pore, and you can see these purple things hanging out. Those purple things are the ones which will target your molecule. So if I have a particular antigen, or let's say a particular protein, then I will have these purple things will be the aptamers. Okay? Aptamers are basically DNA sequences that recognize proteins. Or if I want to look at DNA bases, to see whether a particular base I want to target, I will have the complementary base on these purple strands. The advantage of this approach is that I have the same underlying thing. I can take off this DNA origami sheet, put another sheet, and use the same thing to send something else. Right? So that way it is sort of easy for me to make large arrays. So if I have, let's say, a 100 by 100 array or something of pores, in this pore I can put origamis corresponding to one molecule. In that one I can put origamis for another molecule, and that way I can get something that can do multiple things. We have not done that yet, but that's what we want to go there. So this kind of approach where we combine the DNA nanotechnology along with this one, we call this our hybrid nanopore method. Now, a little bit about DNA origami. How does it work? So how it works is that basically we start with something called as viral M13 DNA. This is what everybody uses. It's a circular DNA. So imagine that you have a very big, this is like a rubber band, right? This is a very big this thing. And then what you do is that you have these very short DNA things that you see here. Those are called staples. So what is happening is that you have a big circle and you're stapling this circle at various places to create shapes. So for example, if you take a circle and then take these two corners and pin it, then you'll get like an H shape, right? So like that, if you pin it at various locations, you'll get whatever shape you want. Now how do you do that? Is basically there's a computational framework to do that. So you can, whatever shape you desire, you can buy this circular DNA, and then you can buy these staples. Like, so the program will tell you these are the staples that are required to create this particular shape. So you can order them from various companies. So hundreds of staple strands. So in this particular case, we use about, I think, 120 or so unique sequences to do this thing. Okay. So this is the design part, and then it will tell you that these are the staples that are required to give you the sequence of these things, and then you buy them. There are companies that will sell you this. So here is how the shape should look like. And here is finally when you make the thing. So you can see that sort of kind of looks like what we have here. There is this little overhang to make sure which side it is lying on and things like that. And here you can see, somewhere here you can see there is this overhang that you can see there. And if you look very closely, you can see in the center there is a small depression. And you can see in the image also there is a slightly darker region in the center. That is because in the center, in this region, we have a very small pore. It's about a 2 nanometer kind of gap that we have here. So all those recognition elements I talked about, the aptamers and things and all that, they will be sitting there. Okay, so and here's the sort of zoomed out version of that thing. So the idea is that you will have this aptamer. It can also be nucleotide sequences that will target its complementary thing in the sample. So again, using this principle, we were able to demonstrate that we could do single protein detection. So here's an example where we have two different proteins. One is thrombin, another is septorin. It doesn't really matter what these proteins are. This is only a demonstration of what is possible. So what we have is the purple strands. Those are the aptamer for thrombin. So that means that those sequences can target the thrombin molecule. They will bind the thrombin molecule. So when you look at this, what you see here is these are like traces which are concatenated from lots of experiments. There are like probably a few hundred of such events. So if you look at streptorin, you see that there's basically the baseline. There's nothing that you see which is blocking, right? So this is the negative blockade. When you put thrombin, you see that you start seeing these blockades. So the reason is because these aptamers are targeting thrombin. So when thrombin comes, it will bind the thrombin. The thrombin sits there for much longer time, and it will block for more long, right? Because all of these are stochastic systems, right? It's not like it comes and sits there and then never leaves. So it comes and then leaves. But the thrombin, because it is attracted by these aptamers, it will stay there for much longer, and therefore, on an average, it creates a much larger blockade than streptorin. So you can see that they are very extremely distinct signals. You can never distinguish them. And each of these spikes is one molecule. And again, how do you confirm that? What you can do is you can looking at the statistics of these things. like there is a, yeah, so if you look at the raw trace, right, so for example something like that, you see that there's a distance between the spikes, that's the inter-spike time, and the spikes themselves have some time scale, which is the dwell time or something like that. So using those two numbers we can calculate what is the kinetic parameters, which is like the reaction rate, K on, K off, and those sort of things. And when you calculate that, they kind of match with what is reported in literature. So that also tells you that, yeah, I mean, kind of confirms that we are actually measuring single molecule proteins. Good. Okay. We can, I mean, maybe I don't spend much time, but we can also do other things, like for example, DNA forms these secondary structures called G polymerases. And so we can also measure that. So for example, in KCl, DNA forms the structure, NaCl doesn't form the structure, and we can actually see the difference. So we can do things of that kind also. And finally, what I want to show is that not just proteins, but suppose you want to detect, as I mentioned, today's solid-state nanopores, they cannot sequence. They cannot recognize the sequences sitting there. So we thought, can we at least do something better? I mean, something at least to an extent where, suppose you have a sequence that has a special region. So in this case, the special region is composed of, let's say, 10 Gs or something. Okay. So can we make a nanopore system where it can recognize that, okay, here is a sequence with 10 Gs. And can it distinguish that from a sequence which contains, let's say, only 8 Gs or 4 Gs or 2 Gs? So this is not getting to really sequencing at single base resolution, but at least blocks of bases can read. So what we did there is that same idea, but instead of those aptamers, we have the complementary sequence. So if you're trying to target the G, what we will do is that we will put a C sequence on the aptamer, and what we see here is, so here is, for example, so here is the sequence. And we have different names. So we G0 represents the sequence where there are no Gs. That's like a negative number. And then G2 is the sequence which has 2 Gs, 4 Gs, 8 Gs, and 10 Gs. Okay. So here is comparison of G10 versus C. So C is basically G0. Like C means that there are no Gs, and you can see that it's a very clear distinction. Okay. So it can recognize that there is a G10. Okay. But what we want to do is that can it also recognize the difference between G10 versus G8 and G4. So we did that, and the main result is here. What we can show is that it can distinguish up to G4. So it can distinguish G10 up to G4. G2 it cannot distinguish from C. But what it tells us that right now we can recognize 4 base sequences. So in the DNA strand, if you say that I want to target this particular thing which is like 4 bases long, today I can detect that. And that is good because a lot of these PCR reactions and so on, they are looking for like a primal region, and then that is how they specifically create this. So the primal regions are typically like 20 bases long. So if we can detect 4 bases, definitely 20 base long sequences is something that we should be able to detect. So the idea that we want to go forward with is that can we make systems where we can do, for example, things like detection of bacteria or specific portions of the genome using this method. And the hope is that maybe we don't need to do PCR, because PCR is a very time-consuming step which is involved in diagnostics, and is one of the very restrictive steps. So that takes 2 to 3 hours to do a full amplification, depending on the various states. So if you can avoid PCR, then we can have very rapid tests which are as accurate as PCR-based diagnostics. So that is where we want to go next. So again, so that's the thing, we can detect homonuclear sequences as short as 4 BP long. The question is that can we detect 12 to BP long specific sequences, like for example primal regions and things like that. And so eventually what we want to do is to do PCR-free detection of pathogens and microRNAs and so on. Because in cancer, for example, people have shown that if you measure the microRNA profile, it can give you lots of insights into what kind of cancer it is, what stage it is, and things like that. So the idea is that can we then start detecting microRNAs are also about 10 to 15 bases long. Right? So we have shown that we can go as low as 4. So 8, 10, 15 should be something that we should be able to target. So these are sort of like the future things that we want to do. And there are also other, as I mentioned, there are some other stories that I have not talked about, which is basically we want to build electronics for the hour, which can do much better than what we can do today. And we do a lot of simulations. We do a lot of simulations to try to understand how these molecules are going. Right, so do a lot of molecular simulation. And then finally, some other things like, for example, if you have a DNA, a single-strand DNA, and let's say that some probes have bound in various places. So there are certain locations that are double-stranded, and some locations are single-stranded. So here we show that we can detect those things as well. These are some of the things that I didn't talk about. Again, these are sort of things that we are still working on, which is like, PCR field diagnosis, integration of ultra thin things that we were talking about. So we want to come up with a method where we can have, let's say, for example, 100 pores, all of them sealed with 2D materials. So right now we have sort of kind of in a stage where we have seen some data where we are able to put these things called MMT, it's like a clay layered material. So we've been able to put 3 to 5 nanometer thick layers on top of these pores. Again, in a very scalable way. It's not like you don't have to go and stamp them or things of that kind. So there's no manual listing. It's like a self-assembly process. And we want to take that forward to graphene and then these things called covalent organic frameworks and things. People make these ultra thin sheets in various methods. So we want to take those sheets and then try to put them in, a system. And finally, again, a lot of data analysis today is based on parametric things. You have to specify like the depth and those sort of things. So we want to go to a thing where you can specify simply by in terms of shape. So basically, you saw the data, right? The data looks it's like a raw data, and basically you have to see a lot of the spike trains. So it looks very ugly and messy. And trying to extract data out of that is not easy. There are some challenges there. So we want to come up to a system where user can say that I want to look at a particular, and again, in the DNA double sampling, I showed you a particular shape where it is deeper, then it comes back to half level and then it goes like that. There's a shape there. So the user should be able to specify a shape and ask the program to pull up all the events which have that particular shape. So that will require some kind of integration with AI/ML models, this large language models that people use, ChatGPT, those sort of things. It will require some sort of thing. Again, we are working on some of those aspects as well. So, of course, there's other data. So that's basically what I wanted to cover. So I hope that I've given you some idea of what the nanopore technology is, and there are different things which comprise there, different types of nanopores. In particular, we are working on solid state, and I also showed you some examples of what we are doing in the lab. So our sense application, we're fortunate to be in sense that we are able to take this nanopore thing all the way from silicon wafer to the complete chip level, and then we cover all aspects of nanopore, including data analysis. So with that, I'll stop and I'll take any questions that you have. Is that they will use a carrier to transport the analyte. So for example, there may be peptides which are neutral. So what they will do is that they will attach, let's say, a long DNA chain to the peptide, and when controlling the length of the chain, they can control the interaction of the electric field with this one. So those are the kind of things that people use to control the weight.
Won't it be a good idea if you use a graphene so that it will get a more nanochannels?
Definitely. So that is one of the, so people have demonstrated some things with graphene so far, where they have taken exfoliated graphene and then put them on top and so on. So far, they have only been able to distinguish between double strand versus single strand. Okay. So they can just say that it is double strand versus single strand. So just putting graphene alone is not enough. There has to be this slowing down and things of that kind which needs to be done. So the question is that how do you integrate both of those things? So that is one of the things that we are working on. And then, as I said, also this idea of taking the exfoliated graphene and then stamping on top is not scalable. You can't make commercial scale production or those sort of things with that kind of technology. So we want to look at something that is scalable. This is in the sense that, so what we try to do is that in our sample, we have these 2D sheets in our reservoir. And when you apply voltage, because of the flow, these 2D sheets will come and sit on top of the nanopore in a very sort of self-aligning manner. So that is the thing that we are trying to optimize and get.
We can screen them, right?
We can?
We can screen them, like if they have some charges.
It's not that easy because again, because there are lots of things that happen, this clumping and a lot of things that happen. So controlling that stability, chemical stability of those things are not easy. So it's not. I have not come across things of that of this kind. Again, filtering, screening, and all that to get the right things and so on. Right now, things are a little bit stochastic, but the hope is that the other thing is that if you have this CVD graphene, monolayer graphene, which you can buy, is also a little bit expensive. But if you have access to large amounts of those, that's how it solves the problem.
Manoj, just continue with this. I just want to know, we can go high throughput with this, right? Like,
yeah.
And what could be the limiting thing for this? Like, high throughput means how fast you can go? Like, what is the limiting
So
factor?
right now, if you look at sequencing, the limitation is basically the speed, electronic, as I said, the kilohertz rate. So people can read 400 bases per second. So that is like the limit. And if you go faster than that, you just don't have enough SNR. But then what they do is that they have many parallel ports, and then you do like basically parallel this thing. So as a result, the effectivity of the report is higher. So right now the limitation is basically the SNR.
Okay.
So the readout, an ionic current based readout has an SNR which is equivalent to about 400 basis per second. So that is why again one of the avenues of research is to see whether we can come up with other technology, maybe for example optics based and things like that. Maybe we can have a higher rate, maybe let's say at least from 400 to let's say 4000 basis per second or something like that can be improved. That is again one line of research that is.
But the limitation that you said is only for one core.
Yeah.
So if you have multiple cores, you can
Yeah.
go to megahertz as well.
But then you have to measure the same thing. You should be measuring the same thing over multiple cores.
Okay.
So the other thing is that can you make pores where you can actually do, so right now all the pores are unidirectional. So molecules can only go from one side to the other side. There's no pore where you can switch the directions. If you can switch it, then the same thing you can measure multiple times.
Okay.
Then it doesn't matter. Then you don't have to do the same measurements over multiple. No, you don't have to do the same thing in every pore. You can do different things in different pores, but then do multiple times and things like that.
Just one more thing. What drives the flow? Is it some kind of pump that will be
Electrical. It's just an
Okay,
electro.
correct. Okay.
But people have tried using pumps and things, but the problem is that because the pore is so small, you require very large pressures. It's like the HPLC kind of thing, right? I mean, so you require extremely large pressures to have decent flow rates. So electric field is the most energy efficient.
Okay. That seems to be the driving factor.
Yes, sir.
Okay. Anyone else want to ask anything? Like,
So the molecule
okay.
vibrate or rotate vibrate or rotate impact or what?
Okay. So the forces are very small. I mean, so those sort of forces are going to be fairly small.
Yes, vibrate or rotate.
Yeah. So one of the things that people have tried to do is that making thicker pores. Right? But then what happens is the molecule comes and then spends a lot of time in the pore. But when it spends, the molecule will be rotating and also it will be doing something which is characterized by rotational diffusion of the molecule, which will depend on the shape and things like that. People have tried to see whether the shape of the molecule and the rotational diffusion coefficients and so on can be determined from that sort of data. But if you're asking about the mechanical impact and so on, there is no such impact. I mean, it doesn't hurt the pore in any way.
So can we use AC pumps to create that bidirectional flow that you are talking about?
So that is possible, but the thing is that in this, effect how far you can switch the electric fields, it basically depends on your capacitive thing. Right? I mean, there's an RC time constant for the system. So that time constant, if you calculate for these systems, it comes to something like maybe again 10 kilohertz or so. So the thing is that it is still not significantly faster than what we can do. Maybe you can get a factor of 5 or 6 from that sort of a thing, but that is not enough. I mean, from one sort of 400 bases, we want to go to like 4 billion. I mean, we want to make very big jumps. So this approach of switching within the fluid field, actually people have not explored that much at all. I mean, I don't think there's any work at all. Nobody has really tried it. And again, part of the limitation is this electronics and so on. Again, it's not a very explored area. But it is promising. It is something that in fact we wanted to do in our group. We want to explore that to some extent in our group. But the limitation is going to be from this capacitance of your system. System is inherently has a very high capacity because all these membranes are there, right? Charges can get attached to them. So they have very large capacitance. So as a result, this electrical response is very slow.
The aptamers that you were talking,
Yeah.
they will go and bind to the pore.
Yeah. So they are not binding. So they are not binding the pore. So we have a DNA origami structure. When we designed the origami, at that time itself the aptamers are part of the origami. So when the origami goes and sits, the aptamers are along with them. So the aptamers don't have to go and bind to the pore.
okay. Anyone else want to ask?
So what are the limitations that restrict us to detect less than 4 molecular atoms?
Less than?
4
Yeah. So that is primarily the speed at which things are going. That is the only thing. So if we can actually reduce that to some extent, we should be able to go down to a single, eventually single. See, we need to get to a speed of 1 base per millisecond. So right now people have increased the viscosity of the solution, reduced temperature and all that. Still it is about 100 to 1000 bases per millisecond. So still about 2 to 3 orders of magnitude faster than where we want to be compared to the biological course. So how to reduce it to 100 times slower than what it is today? That is the open challenge.
We can get more blockage in that.
Yeah, so that is again some of the things that we're working on to try to maybe make pores which have some kind of these constrictive regions where things are much slower, and so Again, fabrication challenges and those sort of things are there, but those are some of the approaches that again we are looking into.
Okay. Good. So if I think there's a lot of enthusiasm and a lot of questions, it's a quite interesting thing. Okay, so if no more questions, then let us thank Prof. Manoj.